You're listening to Let's Talk Creation with Todd Wood and Paul Garner, the creation show where we learn, grow, and worship.
Welcome back to another episode of Let's Talk Creation with Todd and Paul. I am Todd Wood.
And I'm Paul Garner.
And before we get started, just want to remind you all to click that like button, click that subscribe button, click the notification bell. If you're on a different platform, interact with us in some other way than just consuming and listening. Leave a review, share it on your social media platforms, whichever one you're on. We do appreciate all that attention. It helps us beat the algorithm, which is working against us to keep us down. So we do appreciate all that help. Now, before we get going here, I do want to acknowledge, yes, I am not in the cabin. It is pouring down rain outside. It's been storming all night and it's still rainy. And whenever that happens, it's always noisy out there. So that's why I'm here in my office. And if you've been in my office, you know it's a bit of a mess. And it's even worse now with all of my gear set up here. But anyway, we're going to try our best to get through this together, shall we?
Indeed.
Yeah. So another thing before we get started, Paul, there's something coming up here. Not so secret, but we definitely wanted to mention this. This is going to be time sensitive. So why don't you go ahead and tell us what is coming up very soon?
Yeah, very soon. So we're excited to say that on the 19th of June, which is a Friday, Todd is not going to be in his cabin and he's not going to be in his office. He is going to be over here in the UK, visiting us in Cambridge. And one of the things that we're doing while he is here is we are holding a public meeting, Why Science Needs Creationism, at 7:30 at King's Church. There are full details on our new Cambridge House website, which is chproject.org.uk. So you can get the details. So if you are in Britain and you want to come and meet us in person, hear us speak, then we are going to be in Cambridge Friday, 19th of June at 7:30. And we're going to be joined by my good friend and colleague, Dr. Stephen Lloyd. So the three of us are going to be talking about why science needs creationism. So this is your opportunity to come along and meet the three of us in person. So we hope to see you there.
Yeah, that would be great. I would also say, for you folks thinking, hey, this is an expensive time to go internationally traveling. You are correct. It is an expensive time to be going internationally traveling. If you'd like to make a contribution to help us out with traveling expenses, we would very, very much appreciate that. While I'm there, we're not just going to be doing this public event, but we will also be recording. How many did you have on the list? I think like a half a dozen episodes.
Yeah, about 7 episodes, probably.
Yeah. So we're going to do a lot of episodes live together there while I'm over there in jolly old England. And as you can imagine, 2 weeks in Cambridge there in June is going to be an expensive endeavor. And we do appreciate all the finances that people have already given, because this has already been announced. But if you would like to make more contributions, we would very much appreciate that. And
indeed. And we're also going to be doing a summer school. We're both taking part in our student summer school, which is the first one we've ever held, which is going to be in Highbury in London. And so that's coming up very soon too. So there's a lot going on in June. It's going to be a busy time.
Busy.
And we would be delighted if you were able to contribute to help us, cover the costs of that trip, because it's going to be a great time, I think.
Yep. And you can find out how to make contributions to our ministries by just visiting our website, letstalkcreation.org. There is a page there where it will show you links to our different donation pages. There are ministries, Core Academy of Science, or Biblical Creation Trust. We'd be very, very grateful for your support. This is going to be, I think, a really great trip. We're going to get a lot of stuff done. We're going to get so many podcasts recorded. And it's coming at a lovely opportune moment because we're in the midst of finishing up the manuscript on our book. Yeah. So I'm sure that's going to come up as we are together again. We'll probably have to sit down and go over some editorial issues and diagram issues and all that good stuff as we are putting the finishing touches on that. So yet another cool thing to look forward to. All right. Well, I think that takes care of all of our housekeeping stuff. That's why I'm not in the cabin, why I'm in the office today. That is what's coming up soon. Time sensitive, of course. If you're listening to this after June of 26, sorry. You can still make donations, but we've already gone to England and probably come back. Anyway. So now we are back here. Let's talk about this episode. How about that? We're doing another installment in one of our lovely intermittent series. We have many of them going on, where we sort of throw in an episode every now and then on a particular topic. Now, 2 years ago, and I looked this up on YouTube just to make sure that I had this right. 2 years ago, on episode 85, we did an episode in the Natural Evil intermittent series on the subject of viruses. It is a very general description of, just very, very general description of the biology of viruses, and then a little bit on sort of the theological challenge of what viruses represent. Very, very minimal. We talked about, how they look like they're designed, but they're not obviously designed to help us, at least not obviously designed to help us. And in that episode, we promised that someday we would come back and talk about viruses in more detail. And as usual, there's always interesting things to talk about, so it's taken us a while. But here we are. We finally made it back 2 years later. It just shows you there's so much to say in the world of creation research. So let me look at my other notes here. We have with us today a returning guest, Joe Francis. Welcome, Joe. Hello, Joe.
Yes, great to be here.
It's good to see you, Joe.
You might remember Joe from a previous episode on microbes. We will link to that in the show notes. Joe has a lot of experience in the research world on the subject of microbes and little wee beasties and all that good stuff. So he's a perfect guy to help us sort of try to think through the world of viruses. By way of formal introduction, Joe is an emeritus professor of biology from the Masters University. He still teaches online at Masters and also is a professor online with Liberty University. And he also occasionally teaches for a new startup Christian online university for teachers called Graceland University. So he definitely wanted me to put that into our introduction here. So there you go. All right. Well, yeah. Viruses. Viruses are little microscopic biochemical packages of DNA and protein that hijack cells and get them to replicate the virus. So we didn't really talk much in episode 85 about the actual diversity of viruses. And I know that viruses are insanely diverse. We sort of talked a little bit about like single-stranded DNA and double-stranded RNA and DNA RNA hybrid viruses and all sorts of those kinds of things, but we didn't really get into much detail. So, Joe, I thought we would open here. I know you are well known, at least in my mind, for cool things. Cool things about all sorts of things, and I thought, why don't you share with us some nifty viruses and some of the weird diversity in the viral world?
Sure. And I spent some time this morning looking for my Oxford cabbie hat, and I found it, but it's way too hot. I wanted to wear it in honor of Paul. I know he had some losses lately, and but now I realize that I'm wearing it because you're going on a great trip and have a great conference is coming up. So that's great to hear. Yeah, diversity in viruses. I mean, your question to me too was about some examples of crazy and unusual viruses. But before that, when you say diversity, it's hard to understand the diversity because they all have basically several common parts. DNA or RNA, nucleic acid, as we call them, a coat, protein coat, and a lipid envelope. Not all of them necessarily have lipid envelopes. There are some rare ones that don't have the protein coat. That's about it. And so that way they're very similar. So how can they be so diverse? But let me tell you a crazy story. At least 3 of them on, and it'll show definitely some diversity. The first one's a little disturbing because it's about a virus that causes illness. And I'm going to make a case that if we look at the species of viruses and the ones that are forecasted, those that cause illness are actually very rare. Doesn't feel that way when you have that cold or flu, but they're very rare. I want to talk about the chicken head virus. And you're saying,
What?
The chicken head virus.
Chicken
Wait a
head.
minute. Now, wait a minute. That's a little play on words, because in the 1970s, 80s, there was an infection in animals that could cross over to humans in Europe. And it was in foxes. You're going to have to guess the virus in a minute. And they decided it was so bad because it can cause issues in livestock and humans. They decided to drop vaccines with sausages, inside sausages. The problem is the foxes were so smart, they gathered them up, waited a few weeks till everything was dead and cured and the virus had no activity, and ate the sausages. How they knew to do that, I don't know. So they found out that chicken heads have a really strong smell, and they cannot resist eating them. So they dropped chicken heads all over Switzerland and various countries. And you imagine taking a walk in the woods, not only to be rained on by chicken heads, but to see them all over the place. So now, have I given you enough clues to guess what virus that is?
I'm afraid I've
It's I mean, to go to that effort means that it is deadly in humans, one of the most deadly viruses. So think of deadly viruses in animals.
I mean, the only one that's coming to my mind is smallpox.
Yeah, smallpox. What's interesting is smallpox has been eradicated. It's only in 2 places on Earth: a vault somewhere, I think, in Georgia, and a vault in Russia somewhere for research purposes. Good guess, but it's been eliminated. It's hard to believe.
Okay, so this is after that. All right.
Yeah. Actually, the chicken head gives an example because this virus goes to the brain. Here's even what's weirder.
Sorry, I am sorry, but I am not thinking of a brain virus here.
Nervous system, brain. It goes to the brain. Here's what's even weirder. It's deadly to humans by the time it gets to the brain, but it induces hydrophobia. People get terribly scared of water.
Rabies?
Oh, rabies.
Rabies. Yes. Yes.
Oh,
Wow.
goodness.
Wow.
So that's a strange, you said crazy and unusual. But
That
here's
is crazy and unusual.
Yeah.
Mhm.
What the vaccine delivery is strange and unusual. But here's what's even more strange and unusual. I wouldn't have picked it if there wasn't more. And the more is this. It's got it looks like a bullet, by the way.
Mm.
And it's strange that in HIV viruses, the virus so both HIV and rabies have a lipid envelope, then this protein core. The protein core looks like a bullet in rabies. In HIV, it looks like a sarcophagus. And it's just like, that's so weird that we have these shapes that remind us of things that can kill us. But here's where the science comes in to show how strange this one is. It only has 5 proteins. So how in the world does it get to the brain? Because with an animal bite, which is usually a problem, it has to get from the arm or the leg to your brain, and it has to get inside neurons. So think for a minute, how does a virus get inside neurons? Neurons are pretty good outer cover. They get to neurons at the interface between, okay, think about all your neurons eventually end up what? Going to your nervous system, but then going to our muscle cells. So it's at that interface is where rabies gets in. And so now how does it get to the brain? This is so bizarre with 5 proteins. It has one protein that binds to dynein. If you remember dynein and other proteins like this, they're motor proteins.
Yeah.
And they came out a few years ago, and there's videos everywhere because they actually have these little look like little arm pods or legs that by protein mechanics attach to a cytoskeleton fiber, and the other one attaches like they're walking. You've seen the videos. So that's what's so wild about this virus. It has one protein, the P protein. I think it's a phosphoprotein that somehow binds to dynein, and dynein's inside the virus. Remember, cytoskeleton fibers are for our audience, give us our cells shapes, and there can be long rods. So in a neuron, you have a long axon full of these cytoskeletal components, and this motor protein that walks on them all the way to the cell body, lyses the cell body, goes to the. I don't know how the directionality works. It must be so. I mean, how does it go from your arm to your brain? It must be so much virus. It just keeps attacking nerve cells and eventually gets to the brain. By the time it gets there, it's too late. Brain swelling is what causes death, and this strange hydrophobia, and so on. The good news is, I was looking for good news about this virus. The good news is we are mostly vaccinated, but the other good news is it's one of the few viruses where, because it takes 4 weeks to get to your brain. That's the shortest period of time. So you can be vaccinated within those 4 weeks. That's the best part, right? You get bit by a dog. We had our log cabin here. We get bats between our logs outside. And I listen to them about this time of year. I turn a fan on so they don't keep me awake all night. And about every other summer, we get one in the house. I use a fish net. I learned how to get them with a fishing net. But one time, one got in a room. We found it dead, and the cat wasn't there. And I'm like, oh no. Was she bitten? So we called the public health service, and we put the bat in a garage freezer, and they came and got it and tested it and said it was negative. So it is important to check those things if that happens, because you don't know. Did it bite the animal? Could the animal didn't bite you, and so on. But what's good about that is we can be vaccinated right away. I think the original vaccinations, I think they still do it in the stomach muscles, which it's not pleasant. So wild and strange viruses. Are you ready for number 2?
Yeah.
Okay.
I'm still stuck on chicken head viruses, but yeah, let's do number 2 here.
Yeah.
That is some story. So if I understand, they were vaccinating the animals that were eating the chicken heads. That's the idea.
Yeah.
The foxes.
Yes. And the question is, now I'm wondering too. I don't know the full story here, because if that virus goes, it's going to go to the chicken head, but you don't necessarily want live virus because you could kill the animal.
Right.
So what they use in the vaccine is just like the protein coat. I'm not sure how all that, maybe they just feed the chickens the protein coat and do a lot of head chopping. And okay. I hope the other part of the chicken's not in the store, the meat market. Oh man. Mimi virus. This is one of the largest viruses, and it's called Mimi because it mimics a cell. It's so big. They were found in water towers about 10 years ago, maybe longer now. Then they found a larger version, so they call it the mama virus. So there's a Mimi virus and the mama virus. they infect amoebas, and in these water towers, they found them infecting amoebas. They can infect our white blood cells called macrophages. There's a debate whether they can cause disease. Some people have said, well, we keep finding them in pneumonia patients, but they don't know. Here's the wild stuff. It's so big that it interfaces with other viruses that get inside it. These are called satellite viruses. And, or this one's actually called the satellite virus. I can say the one they originally found had this architecture that looked like a space satellite, one of the Russian ones called Sputnik. So the first virus I think they got from Mebi virus, they call it now the Sputnik virus. Here's the weird thing. Inside the Sputnik virus, we find proteins and genes from archaeans, eukaryotes, and prokaryotes. That's bizarre. That's bizarre. The three, for our audience, the three worlds of creatures that based on their cell type.
Yeah.
So eukaryotes, we are eukaryotic cells. We are eukaryotic individual people or creatures. Prokaryotes are the bacteria, and archaeans are a kind of bacterium called extremophiles. We find them in hot springs and things. And so those are considered to be the three domains of life. Sputnik carries all three. That is bizarre and kind of worrisome. I think, what's going on with this interface of the viral world with three of the major domains of life. It performs lateral gene transfer to all three, so it's trading genes. Okay, that's another strange one.
And you kept saying, here's the interesting part, and everything that you said was interesting. So just to clarify here, when you said Mimi virus, okay, that is as big as a cell and mimics an actual cell. You're not kidding, right? So this thing has so a bacterial genome might have a big one might have 10 to 20,000 nucleotides
right
in the bacterial genome of a virus. Mimi virus has 500,000, if I remember correctly. And Mama virus is even bigger than that.
Yes.
That's incredible. And then they have membranes that look like cell membranes, but they're viruses. And then they have viruses that infect the cell membrane that is not a cell membrane on the virus.
Yeah.
Virus is all the way down, I guess.
I was wondering too, apparently the membrane, oh gosh, I think it's inside the protein coat. So it doesn't really have a cytoplasm. It's missing that component of a cell.
Well, yeah, because it's not doing metabolism of its own. It's just looking to hijack something else. Okay.
On the microscope, it's very roundish, like a small, bacteria.
Wow.
Yeah. The third weird one was actually the most fun, and it's probably the most common in virology textbooks, is the T4 lander. That's a phage.
Oh, yeah.
And phages attack bacteria, so they're called bacteriophages, but we'll just call them phages from now on. And they exclusively attack bacteria. We don't find any crossover. Sputnik is a weird thing, but we don't find phages attaching to our cells and trying to get in. Many phages look like spaceships. They have this icosahedral shell, no lipid envelope. Many are called tailed. They have this tail and then tail fibers. Okay, here is the crazy unusual stuff. They can walk across the bacteria. Okay, those tail fibers. So apparently, this would be a beautiful study. I'm sure on the end of those tail fibers, they have some kind of molecular structure that binds to surface of the surface proteins that are sticking out, but only binds to one really good, and that's a receptor that they use to get into the cell. So apparently there must be, just slight binding to these other, because otherwise they're going to float away. There has to be binding, and it's not strong enough so that tail just breaks off and kind of moves around, and it just keeps walking until looks like it's walking. It's random, and it finds that protein. One was lipopolysaccharide, I think. These are exterior proteins sticking out. And here's the weird part. It will then, I don't think they know how this works. That tail fiber, it kind of looks in drawings like a spring. It actually compresses and starts to push that round tail into the coat of the bacteria. Now, if you study bacteria, they have a pretty strong coat on the outside. They have their membranes. There's layers outside that membrane that are filled with proteins and other things, and protein lipids, weak proteolipids that stick up. It's thick. The bacterial cell wall and membrane is very thick. So they've actually done some kind of microscopy here, one of the electron microscopy procedures, and they've actually noticed that it actually rotates. It drills through the outer layers. It actually rotates. So it compresses, and that portal actually, it just twists around and gets through those outer membranes. I had no idea until just studying this yesterday that they actually did the drilling part. And that's how the DNA gets in. It's called the T4 lander because it reminded us of a spaceship landing on something.
Yeah. I remember, this is a classic thing in biochemistry and viral studies. They show you microbiology, whatever. It's time to talk about phages. Look at this one. It looks like a lunar lander module that's going to land on the moon. I had no idea those legs were actually legs. I just thought that was a cute analogy, kind of a coincidence, but apparently not.
Yeah, it's called tail fibers, but it might be, you say, why doesn't it float away? But I hope to in our conference in a few weeks talk about that. But the surface binding on the bacteria can be crowded, and my guess is they're just held there by associations with their friends that are trying to get in too, and they're all walking around, kind of making a cap on that cell. Those are my three weird and usual ones. So we can go on to the second topic if you want.
Those are wild. That is amazing. I mean, I didn't realize rabies was so small.
Yeah.
Yes. I mean, you're talking. It's infamous, right? So we always hear about it. You don't want to get bit by a rabid dog or whatever. But yeah, I had no idea it was doing that chaos with just 5 proteins.
Yeah, in good point. It's physically small because I imagine a neuron, those extensions on the neuron are very small too, so they have to have room to get through.
Mm-hmm.
Yeah. And then I think our second topic was complexity of viral functions.
Yeah, I was gonna ask you about that because in the previous episode on the virus subject, I had made the claim that viruses look like they're designed, that they have such amazing complexity, even with, I mean, even in rabies where it's able to have just the right 5 proteins to infect the nervous system. That's a pretty amazing coincidence, if you will. So yeah, how about we talk a little bit about the complexity of viruses and how that works.
Yeah, and I don't mean to get into the really evil ones, but HIV virus has a lot of complexity. The HIV viruses and they to do so much damage to actually and they get into only one type of immune cell basically to destroy that immune. So it's just targeted. The complexity right there is targeted. We have a lot of immune cells in our blood. We have the phagocytes that eat, and there's, oh gosh, maybe 10, 12 classifications of those. Then everyone has heard about the lymphocytes or the T cells. There are several versions of those. We have helper T cells and cytotoxic T cells. So the helper T cell basically says, I see something foreign. I'm going to call over the cytotoxic T cell, which extrudes all these enzymes to kill whatever it's looking at. The HIV is directed only to the helper T cell. That kind of looks like design, right? If you're going to shut down the immune system, you go right to the primary information carrier, the guy who's going to alert everything, and that's the one that kills. So I was in a hospital in Detroit working as a lab tech and working my way through grad school, and I was working with white blood cells for a doctor there, looking at white blood cells from patients in the kidney area of the hospital. And he came in one day and said, put your gloves on, put a mask on. And I had a fluorescence microscope. And in the fluorescence microscope, what you can do is you can look at different cell populations by putting a fluorescent tag on them. So I had a fluorescent antibody to the helper cells, CD4, and I had a fluorescent antibody to the cytotoxic cells called CD8. HIV, I told, we talked about HIV infecting only one kind of T lymphocyte, one of those white blood cells that fights infection. It happens to be the one that alerts the immune system. And so in the early days of HIV detection, there's a ratio of the helper cells and the cytotoxic cells that it communicates with. So cytotoxic means kill. So those T cells kill things. So the helper cell calls that one over. I got something. There's a given ratio in the blood, like 2-to-1, 2-to-1 helpers versus cytotoxic. If that ratio changes, something's going on. And when I looked at this sample from an HIV patient, the helper cell population was way down. The ratio was off. And that's how we figured out if that damage was going on. And that was spooky, and it was scary. So the complexity is this with the HIV. It's an enveloped virus. It attacks that one T cell by using two receptors. So this is really interesting. It has to have two receptors that it engages with on our T cells. One of the receptors pulls it closer to the cell. The other receptor pulls the HIV into the cell. It turns out that some people, some very famous people, became HIV positive but never got the illness. And that's because they have a genetic condition where they're missing that second receptor. That's kind of interesting. Why does it use two receptors to get in? Is there some complexity or design feature there for what this creature was meant to do? We don't know. More complexity. Once inside, it is an RNA virus. Once inside, it uses a reverse transcriptase. So a transcript is something means copy, right? It uses an enzyme to convert that RNA to DNA and degrades the original RNA. Now you're getting some complexity. You got enzymes here to do this. And synthesizes a second DNA. And now you have double-stranded DNA. That's a lot of complexity right there to make this work. Here's what's even more interesting. That process is error-prone, meaning you get lots of different DNAs that you're going to eventually package into viruses, meaning what have you done there? You've caused more HIVs, viruses to be produced that could thwart the immune system. And if one HIV in the population, we start to become immune to it, we're going to shut it down, its population. It's creating more populations that could infect cells, more diversity. That's wild. That looks designed to have this error prone. Then the large complex navigates through the crowded cytoplasm, enters the host nucleus, and then slices the host cell DNA up, then makes new proteins. But here's what's interesting. When it makes the proteins, they're all attached together. Why? Why didn't they finish the job? Of all the proteins, as they get ready to package these new proteins that can cause infection, they're all stuck together. The final thing is a protease. A protease means chop up protein, and it chops up those segments, those protein segments that cause infection. Now they can work independently. That's where one of the HIV drugs comes in, an antiprotease. So to help people today with HIV, we have 3 drugs, and that one has been effective. But notice what I just said too, the virus is changing all the time. So will there come a time when there'll be a variety of virus that resists that, those things that could happen, the drug that prevents the protease from working? That's just the surface of complexity in HIV.
Yeah.
Here's a summary. Unlike simple viruses that just inject material and hijack basic replication, HIV must carry its own biochemical toolkit, reverse transcriptase, making the DNA, protease, and others. It bridges the RNA and DNA worlds and actively rewrites the genetic rules of the host to establish a lifelong latent infection. So we can certainly move on to next segment, which was about.
Yeah.
Go ahead, Todd.
I think you've raised some interesting points here talking about HIV. I mean, this is, if I have a complaint about design arguments, this would be the big one right here. People talk about complexity indicating design, and yeah, that's nice. This is a pretty sinister design, though. I mean, it's not here to make my life easier or to make other people's lives easier. It's destroying us.
Yes.
And I think, we're trespassing into the world of theology, which
Yes.
of course we would, because we have to. But yeah, I mean, people, want to talk about design and complexity and beauty and wonder, and is this evidence of design? And I think, no, it's terrible. It's not a beautiful thing. It's not a wonderful thing. It's awful. So, and that really sort of reflects, I would say, our attitude toward viruses from the last episode. I have a dim view of viruses. They seem to be designed to get into cells and hijack them and mess them up and kill. And but I know when we talked to you previously, I mean, we were talking about your views on microbes and how microbes kind of get a bum rap, right? It's not fair for us to look at the disease causing things as representative of the whole class of microbes. Do you think your benign view on microbes extends to viruses? Is there a world where viruses are not terrible?
Yeah, and let's talk about that. That's a good question. I share where we're at in creationism. We don't have great answers here, but we do still see design that could have been stolen, or I don't know. But let's go into some really interesting things that are happening with viruses. Some good things, and compare and contrast. It doesn't negate the evil viruses that are still there, but there's some interesting things. One is to think about how many. Well, we can talk about the biomatrix. And I wrote a paper for the ICC on that years ago, and Kurt was my advisor, and he, well, he was the reviewer, I should say. And he wanted to change the name to organosubstrate. I said, okay, because
Right.
he's brilliant and I can't refuse that. And,
And we're talking about Kurt Wise here, just
right.
in case anybody's
And
wondering.
so no one uses that. We all say the biomatrix. What is the biomatrix? It's an idea that microbes are everywhere, and they can be supporting life. And let me give you some examples of the biomatrix, and maybe we can think about why these other viruses, did they come from the biomatrix? How did they go wrong? Are they in a separate class by themselves? Let's talk about a three-way symbiosis of plants that live near hot springs in Yellowstone Park. My wife Becky and I were just there for the first time last September, and I think I could go there every day to photograph animals and things in the park. Well, they have some really nasty hot springs, and we cringed at people trying to walk around them closely. And but there are plants that live right on the edge of those, and they found out that they have a fungus that actually helps them tolerate, near temperatures that boil water. That's how close they are. And they found out that the fungus doesn't work unless there's a virus that's infecting the fungus. That's interesting. You say, well, the fungus is, why isn't the fungus just designed to do that by itself? Well, we're also talking about a certain area of the biomatrix in a certain area. What viruses are good at are just traveling through the air and water. And so maybe they're this messenger that just kind of makes sense that it can be available to that fungus. It's available to all funguses, but maybe those in cold climates reject it. I don't know, but it is an interesting symbiosis. The plants are called tropical panic grass. I don't know why they're called panic, but maybe they're upset about being invaded by so many creatures.
Let's panic.
Let's panic. Yeah. That's limited. That's in one spot. Here's where I think is a really good example of the biomatrix at work and viruses being involved. Todd and I are from Michigan, and they have taken satellite pictures of Lake Erie, I think late summer, and it looks totally green, or at least in one part, you might see blue in one part of the lake, and then you see this stringy, this a new age painting kind of part, which all green. Those are cyanobacteria, which, might be dating myself here, but in the early days of teaching microbiology, 30 years ago or so, they were called blue-green algae. But now everyone was wrong about that. They're bacteria.
Cyanobacteria. Yeah.
Cyanobacteria. And they're very good about photosynthesis. But they can overproduce, and that can start to use up some of the oxygen, even though it's producing it, and so on. So we think there are two major cyanobacteriums. I'm not going to try to pronounce their names, but those two, among many others, we believe produce 45% of the world's oxygen. If you can see these blooms from space, there's a lot of cyanobacteria. And guess what? They are invaded by a cyanophage. Okay, a cyanophage invades the cyanobacteria, breaks it apart, kills it. But what are we doing here? What's going on? Well, there's a lot of carbon there. And you don't necessarily want that carbon going into the atmosphere. So it goes to the bottom of the waterways and causes a sink, okay? A sink for carbon, a carbon sink, which kind of makes sense. That helps the atmosphere. That's a lot of carbon going into this carbon sink. Here's what's even more bizarre. Dicyanophages carry genes for photosynthesis. Okay, they carry genes. They're not really good at it. They don't have a cytoplasm to put all this together. What they have discovered is when a cyanophage infects a cyanobacteria, some time goes by before it breaks open. Now think about this. why doesn't the cyanophage want to break open that cyanobacteria right away? Or how does it break it open? It makes copies of itself. That's pretty much how viruses break out. It just gets so many and then the cell can't. Well, that could take hours and days. This comes from botanists in the field who discovered this. And they said, yeah, it goes into the cyanobacteria, and a lot of them have been sunburned, and it replaces the photosynthetic machinery for those hours or days that it's reproducing itself, and then it dies. So now you have a cyanophage contributing to that making of oxygen. So they think 5% of the world's oxygen is contributed by this virus. Just because it's
That's wild.
World. What in the world?
So that's a fascinating story. It helps.
Yeah.
It's so if you go on and read these environmental microbiologists, yeah, they're probably doing this with lots of different chemicals, cycles in the ocean. The ocean's amazing. There's a phage researcher at one of the top-notch Southern California schools, I think San Diego, who has looked at some of the phages in the ocean, and he says they all have genes from other things. It's like they're a huge vault of genes in the ocean, in these phages. So you've got a lot going on in ecology. And I think part of the answer about design here in God's world is these creatures are recycling things, and are doing it in a way to lower toxicity, because anything can be toxic at a high amount, right? So this is pretty cool.
Oh, wait. This is backwards, Joe.
Yes.
Viruses are making things toxic. Rabies and
Yes.
HIV, it's terrible. And
It
you're
is, yeah.
telling me that actually viruses are reducing toxicity.
Yes.
Okay.
Yes.
All right. That's going to be a difficult one for my brain to latch onto, I think.
Yeah, it's still a conundrum. But major processes going. Yeah, it would be more cool if a cyanobacteria was the HIV virus. Yeah, it's at the fall it started doing the bad things, but we just don't have that evidence, right? We don't have that.
Right.
It's all different. So yeah, the ocean, the viruses in the ocean, we've described 16,000 species. We've documented 16,000 species of viruses. They think there are trillions of species out there. And 2/3 of those species are phages. So, but let's go on to another one, because as I started looking at this, a lot of viruses are involved in immune function, for good or bad. So here's one about immune function that has a good story to it. So we're probably all too young to remember the chestnut trees that grew across the United States in the late 1800s. I mean, as big of trunks as some redwoods. They say they were glorious across our country. It was a fungal blight that basically wiped out nearly all of them. Ecologists were really down in the mouth, except they found some survivors. And what did they find in those survivors? A virus attacking that fungus called a hypovirus. Now the chestnuts are back on track. And so here's a virus. So yeah, this seems like a biomatrixy like function. We could say that it's still killing something, but it's involved. And I see a common theme about viruses being involved in the immune system. We'll get there on that. Let's see. And while we're talking about any thoughts on the chestnut tree before we move on? Pretty amazing.
No, you better keep going, because this is fascinating.
Okay. So on that theme of immune system, we see the HIV destroying a good part of the immune system, but we have endogenous retroviruses are transposons that could be once retroviruses. So they're called HERVs, human endogenous retroviruses, in our DNA. And we know of one that's involved in expressing proteins in the trophectoderm. The trophectoderm is the placental tissue that is growing between the mother and baby. It's that connection. It's all the tissues, and it's part of the tissues involved in that mother to baby connection. We know now that an endogenous retrovirus, an HERV, expresses proteins exclusively in the trophectoderm. And what did we say about HIV? It turns off the immune system. It suppresses it. That's what this virus does. It suppresses some of the proteins that could turn on the immune system, specifically in that trophectoderm. And that's the result of an endogenous retrovirus. And if I had any common theme, it could be the viruses were designed to interact with the immune system. Go ahead, Todd. I think you were.
My brain is being blown here. All right. So, I mean, it's sort of forcing me to reframe the way we think about viruses, because viruses are basically at the core cytotoxic. They're killing cells.
Yes.
Individual. It happens to be that I am made of cells. So sometimes the viruses can make me ill and sometimes they can kill me. And that's bad. Presumably. But killing individual cells by themselves is not bad. Now, one of the things that I know about, and this is research that has been going on. I don't know if you wanted to talk about this, but I'll just throw this out there. This whole issue of oncolytic viruses, viruses that have a strange preference for going after cancer cells and can reduce the size of tumors, because they kill cancer cells more than they kill healthy cells.
Yes.
Utterly fascinating to me. So it seems like the bad rap that they get, their bad reputation, is growing out of this whole issue of selection bias. We're going into the viral world like we go into the microbial world, going, why am I getting sick? What microscopic thing is making me sick? And it makes us think that all microscopic things are just there to make us sick. And in fact, that may not be the case. There's more to it.
Yes.
Yeah.
Yeah. I ran across oncolytic viruses. Don't know much about them. I think they're actually using them in therapy.
Wow.
That's pretty exciting now. They're getting to that stage. But let me give you another example of fighting the immune system. But it's something that Darwin saw too. You know about parasitoid wasps invading caterpillars. I think was it Ichneumonidae his problem? That critter. And well, now we know that while they're injecting their larva, they're injecting a virus to turn off the immune system of that caterpillar. So there's that immune system, and it's an evil one, but that's involved with. It's a common theme that just keeps. I'm an immunologist by training too, so I'm biased, but you keep finding these stories. So I don't know if that would help Darwin to know that virus is injected. Oh, well.
I think he'd find it as a way of fighting back, right?
That's right. That's right.
Yeah.
I don't blame him. That's hard to see in some ways. In our last segment here, though, I'd like to talk about, if there's time remaining, the human viro, because it's really, really interesting. And then some medical things we're using viruses for that are saving lives. Are we good to go on that? Keep going.
We have a couple of minutes left, but maybe 5 minutes. Yeah.
Okay, quickly then. What I found in research, and I presented this at one of our summer conferences a few years ago, is that the microbiome in us contains bacteria and viruses. We know more about the bacteria, it seems like, than the viruses. And part of that reason is we can't find many animal viruses doing good in the body. In fact, our immune system pretty much wipes out all the animal viruses that we're infected with. We carry about 9 or 10 for our lifetime, like herpes virus and things like that. But this is, when you think about the biomatrix of the body, it's bacterial, it's heavy on the bacterial side, except that each one of those species of bacteria, the 700 species that could be living in human guts, each have a phage. And that means, believe it or not, viruses are the most numerous creature living on us, because we believe there's one bacterium for each of our cells. So we made of 35 trillion cells, is it? 35 trillion cells, the human body. There's at least one bacteria for each of those. They're thinking now, they thought it was 10 to 1, now it's about 1 to 1. But then each bacteria can have multiple viruses. So that's interesting. What are all those phages doing? If you come to our conference this summer, I hope to be talking about that. The abstract's accepted. And but that's interesting. But lastly, this. We're using medicine. Let me give you one example. And we're using viruses in gene therapy in medicine. Let me give you an example. There's a muscle deficiency problem called AADC. Children are born with it. The brain stops making neurotransmitters. What is the main thing that our nerves do? We just talked about earlier. They wire our muscles. Those kids just go into paralysis, and the parents have to watch this for 5, 6, 7 years, 10 years. Just about 10 years ago, Taiwanese girl living in the US, her case got in the news. We didn't have US approval for this method, so they did surgery, I think, on 6 children in Taiwan. And what they did was they went into the brain, drilled a hole in the brain, went into the putamen part of the brain, very small area where neurotransmitters are made. They put a gene in there. But if we put a gene in by itself, it would do nothing. They used an adenovirus to do the work. And these, I think 5 kids survived, and now their survival rate's even higher, and started within days moving their muscles. This gene therapy is going on, and it's mostly going to involve viral carriers. That's something good. It's not something natural good. We are involved in it, but just thank the Lord for the tools he has given us. One more thing, and that is, we've heard of the CRISPR system. This CRISPR system is being used also for gene therapy, because we can just take that system from a bacterium and we can put that slices DNA up and can allow us to insert genes. That CRISPR system is only there because the bacteria use it as an immune system against phages. So here you have viruses driving the formation of this CRISPR system, or driving the creation of it, or whatever. And now we're using it to save lives. And, there's a story a few years ago about a Chinese researcher. Remember I talked about the 2 receptors for HIV? He found out that he could modify embryo cells and turn off that one receptor using gene therapy, using the CRISPR system on embryos. He was sent to prison because he had no authority to do it, but that's on the horizon, curing illnesses in children before they're even implanted in the womb. Do we dare go there? My point is, again, virus biology has been used for all these technologies, and I think they're going to be used to save lives, more lives.
Well, that's amazing. As always, I always love talking to Joe. He's got such a shocking perspective on everything. Thank you, Joe, for being with us. Paul, any closing comments from your end?
Well, I mean, I don't know anything about viruses. It's so way out of my sort of comfort zone. But like you, I've always been interested to hear Joe talk about this subject because he's always got these interesting examples of just fascinating things and amazing sort of insights into, that might just give us a little window into what these things were doing before the fall, before the curse. And I just find it mind blowing.
Yeah.
Thank you.
All right. Just got a weird pop-up on one of my computers here. Anyway, sorry about that. I would want to mention here before we go, Joe mentioned the conference coming up this summer. That is the Origins Conference. We are definitely having it. It's at Masters University in Southern California. You can find out more information on that at coreside.org/origins. We would love to have you there. Tickets are available. We still got a month out before the conference happens. So check that out and sign up and join us. We would be happy to see you there. Also remember, I'll be in Cambridge this weekend. As of the time this airs, we have Why Science Needs Creationism coming up 7:30 Friday night, 19th of June. Where is it at?
King's Church.
King's Church
on Tennyson Road in Cambridge.
All right, there we go. So if you are in the Cambridge area and you would love to see Paul and Todd live, well, we'll do our best to not disappoint you. We have such self-deprecating views, don't we? Thanks again for watching us this week. Thanks again to our guest, Joe. This has been a great conversation. We will be back again in 2 weeks with another show. And until then, we will see you later.
See you then.
Thanks for listening to this episode of Let's Talk Creation. For more information, visit us at letstalkcreation.org, where you'll find an archive of past episodes and all our show notes. If you'd like to leave a comment or make a suggestion, you can find us on all the major social media platforms. Let's Talk Creation is brought to you in the U.S. by Core Academy of Science and in the U.K. by Biblical Creation Trust. As a listener-supported ministry, we are grateful for all of your financial support. Find out how you can make a contribution at our website, letstalkcreation.org. Also remember to like, subscribe, and share this episode with your friends. Thanks, and see you next time.